Hello everybody and welcome back. So far we have completed 3 modules. Module 1 was about the structures and characteristics, the chemical structures, characteristics, some other properties of the nucleic acids, proteins, peptides as well as the lipids. In module 2 we have done some organic synthesis, the laboratory methods or the industrial methods for the synthesis of the nucleic acids and their derivatives. On the other hand, in module 3, which was the last previous module, just we have completed that, was about the biology and doing in vitro studies or doing in vitro methodologies on DNA replication and how can we amplify DNA using the PCR polymerase chain reactions, and then finally how can you sequence a DNA. That was about we have talked about 2 sequencing methods. One was the Sanger's method, Sanger's dideoxy method, and the other one was Maxam Gilbert method. So far we have completed these 3 modules, and I think we have covered quite a good part of the chemistry as well as some biology, and so far we have not done any problems solutions. So today I thought that I will talk about or we will discuss about some problems, numerical problems, and then followed by later on synthetic problems, and then the solutions also we will discuss today. But before that, I would like to clarify something which was in the last lecture in the Maxam Gilbert method, the gel electrophoretic diagram. There was some mistake in it. So, I would like to clarify it today now that our target, I think I have found out the issue. Our target DNA was this one was our target sequence, which was 1, 2, 3, 4, 5, 6, 7, 8, total 8 number of nucleobases, and here we have done 9. So, that was the problem, this one should not be there, we are until 8, and this issue came because I had written the full length of DNA, which is not a cleavage part. This is present in all the other vials. So, this will not come up for consideration. So, as you see, the fragmented patterns are stopped here in vial 4, and then we are good, everything else is fine. So, only thing is that since we have 8 nucleobases long DNA, this 9 will not come. So, problems and the solutions. I will discuss about the very quite easy problems, actually not very difficult to do, and some of them might appear in your assignments as well, not quite the same, but closely related. So, first one, question number 1 is, to start with a very easy one, what is the length of a double stranded DNA dsDNA of 500 base pairs. So, if a double stranded DNA has 500 base pairs in it, which means the DNA is 500 mer long, then what will be the length of that DNA? Of course, this is assuming that DNA is straight in nature or the double helix in nature. So, this is a very easy problem, actually you can solve it very quickly, I guess. So, the difference or the distance between 2 nucleobases in a given DNA, if this is a DNA, I am just writing the straight, if this is base 1, if this is base 2, then this distance is, as we know, is around 3.4 angstrom. So, therefore, your answer will be 3.4 multiplied by, you have 500 nucleobases long, which means 500 minus 1, if you want to be really accurate, that many number of distances will be there. So, this is about in angstrom 3.4 into 499 angstrom, or this will come as 1696.6 angstrom. That will be the length of the DNA, which is almost close to 169.66 if you want to convert it to a different unit, nanometer. You can convert it to any other unit also, micrometer, like that. So, that is how of course, you can calculate the length of a DNA. Of course, if you say that length of a single stranded DNA having 500 bases will be the same as this. So, base pair we generally use the term base pairs to mean that you have one base and alternate to the other base. So, we call this as a base pair, and this is the usual terminology which is used when defining a double stranded DNA, or when defining a single stranded DNA we just call bases. Okay. So, this is your first problem. Question number 2. Calculate the concentration of a single stranded DNA which has the sequence 5 prime CGATT CT AG C and 3 prime. This is the sequence of the DNA. If absorbance at 260 is observed to be 1.5 of a ten times diluted sample, or you can tell this as with a dilution factor of 10. Using a cuvette of 1 centimeter with cuvette of 1 centimeter diameter. Given epsilon value, molar extinction coefficient at 260 is close to 92900 liter mole inverse centimeter inverse. So, this is the question that you are given a single stranded DNA which has this sequence, and when you are measuring the UV absorbance at 260 nanometer, you have used a 10 times diluted sample from the parent DNA stock. You have from the stock solution, from the mother single stranded DNA solution, you have done a 10 times dilution, and that 10 times diluted sample was used to observe the UV absorbance, and that UV absorbance shows 1.5, the value of 1.5, and you are using the cuvette which is 1 centimeter path length basically. The epsilon value is given. So, question is what is the concentration of the mother single stranded DNA. So, basically you have to calculate the concentration, and in order to calculate the concentration, what is the basic equation? Beer-Lambert law, that is absorbance at 260 is equal to epsilon C and L. Epsilon is the molar extinction coefficient, which is given here. C you need to calculate. L is the path length or the length through which the light travels, and this is given as 1 centimeter. It is basically the cuvette you are using, that same diameter of the cuvette will be your path length, because that is the length or that is the distance that will be covered by the light. So, and this A value is given as 1.5. So, 1.5 equals to 92,900 into L is 1 into C, concentration. Now, what is the unit? Liter mole inverse centimeter inverse into the length was in centimeter. So, this cancelled out. So, your concentration would be 1.5 divided by 92,900 mole per liter, and that is coming as 1.6 into 10 to the power minus 5 mole per liter, which is equivalent to 16 micromolar. So, mole per liter is molar, capital M. Micromolar is 10 to the power minus 6. So, this would be 16 micromolar, and that is the concentration of the 10 times diluted sample. So, this basically is the absorbance of the diluted sample. So, therefore, concentration of the parent DNA stock, I am writing stock single stranded DNA, that would be 16 micromolar into 10 times or 160 micromolar. This should be the answer to this question. So, answer basically is 160 micromolar. So, very quickly we have used this sequence CG ATT CT A G C 3 prime. Now, if I ask you to calculate a molecular weight of a DNA, how you are going to do? Sometimes you have a very long DNA, of course, and usually you do not have to do anything. You can just type the sequence somewhere. There are many places in the in internet, or there are Excel files also available, where you can just type your sequence and it will give you the molecular weight. So, ideally you do not have to calculate it by hand, but I would like to show you, or I would like to discuss how you can calculate it if you really want to, or if you need to, it's good to know. So, what usually is a practice that when you have a long DNA, then it is assumed that they are composed of all 4 kinds of nucleobases, and in general people think, or in general it is considered that the average molar mass of a DNA, I will write monomer. So, we are calculating the molecular weight of this DNA strand. So, the very quick calculation is that it is considered that the average molar mass of a DNA monomer is approximately 330 gram per mole. That is the number which has been calculated taking the average of all 4 nucleobases, and it is assumed that your DNA will be composed of almost equal number of all 4. So, it is of course not the accurate measurement, but an approximate measurement, approximate calculation. 330 gram per mole is the average molar mass of a DNA monomer, that includes the phosphates, that includes the sugar. So, therefore, for this single stranded DNA, so molecular weight of single stranded DNA, this particular DNA will be, how many bases you have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 into 330 gram per mole. So, approximately 3300 will be your approximate molecular weight of your given DNA. So, this is for the single stranded DNA. If you have to calculate the molecular weight of the double stranded DNA, if this DNA comes in a double strand form, then it will be simply the double of it. So, 10 into 660 gram per mole, that would be the molecular weight of the double stranded DNA. This is for very quick calculation and very approximate calculation. This is not at all close to the perfection, of course. Now, if you really want to calculate the individual, taking into consideration about the individual nucleobases, then you can do that too. So, I will give you a chart which you can use later. So, this is the base, nucleobase, and this is the molecular weight in gram per mole. Now, if you have adenosine, AMP, adenosine monophosphate, then its molecular weight is 331. If you have CMP, cytosine monophosphate or cytidine monophosphate, that has a number 307. GMP is 347, TMP is 322 gram per mole. and this the structure is of course this, which here is the base and this is monophosphate, means O minus OH basically. Now, if you want to calculate using this table, if you want to calculate the molecular weight of the given DNA, then what you have to do? Basically, you sum up of all these molecular weights of the individual bases, but that will not be accurate, because when you are doing the phosphodiester bond formation, OH of the other base B2 is coming and they are doing this reaction. That is why you are getting the phosphodiester. So, it is a minus water molecule is released, one water molecule is released, which has the molecular weight of 18. So, you have to sum up of all the individual nucleobases in terms of the monophosphates, and then minus that many number of phosphate bonds that are there multiplied by the 18, that will give you the correct result. So, in this case, if I calculate molar mass of your single stranded DNA, that would be so 2 into how many adenine is there? 2 into 331 plus cytosine 1, 2, 3, 3 into 307 plus G 1, 2 G 3, 4, 7 plus T 1, 2, 3 T's 3 into 320 minus how many phosphate bonds? Total number of bases minus 1. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and there was this. So, in general formula is n minus 1 into 18, number of bases minus 1. So, if you calculate in this case, so if you take that into consideration, your total value will come as 3081. Yeah, gram per mole. So, here you have the phosphate. So, here nothing is negative, minus 1, 2, 3, 4, 5, 6, 7, 8, 9. So, this would be 9 into 18. So, 10 minus 1, n minus 1 is your now 10 minus 1. So, 9 into 18. So, this would be your the calculated molecular weight. Well, only thing is that you have to remember this table. Similarly, there is you are seeing the epsilon value, that nowadays we just type, if you just type the sequence, it will give you the value of the epsilon of the molecule. But you can there is a way to calculate also. It is little bit complicated and to some extent is boring, that is why I will not show it, but you can, maybe if you are interested, you can go through it, how to calculate the molar extinction coefficient of DNA. So, now question number 3. In order to radioactively label a DNA but not RNA. During replication, then the question is which monomer is appropriate to use. So, suppose you have a mixture of DNA and RNA in your vial and there is replication going on. Now, you want to track only the DNA and therefore, you want to label only the DNA with the radioactive P32. So, okay. So, radioactive label means we are labeling, we are using P32. So, you want to do it selectively for the DNA and not the RNA. Then the question is, I will give you the options. You have to pick up the right kind of monomer that you will use in order to get the labeled DNA. Option A is D ATP with a label P32. Option B is D GTP, C D TTP 32 labeled, and D is D CTP. This last one D UTP. Now, out of these 4, which monomer you will actually use to tag the DNA selectively and not the RNA. So, of course, it is easy. If you just think of replication, now when there is replication going on, what you need? You need polymerase, you need the primer which is there, and you need the free DNTPs. DNTPs would be picked up suitably and the extension will happen. Now, your DNA is there, target DNA is there, target RNA is there, both are having the amplification, which essentially means that you have a DNA polymerase, you have the RNA polymerase also, and all these DNTPs. Now, you find out the monomer which is exclusively used for DNA and not RNA. So, if you start from the top, ATP is a monomer for DNA. Of course, DNA has adenosine, RNA also has adenosine. So, ATP would be taken up by both DNA and the RNA during the replication. So, this cannot be, it will be common. If ATP is taken up, then both DNA would be labeled, RNA will also be labeled. GTP, guanine. Guanine is present in both DNA and RNA. So, similarly, if you use this, then both will be labeled DNA plus RNA. So, this is not the selective one. Thymine, TTP. Thymine is present in DNA, but not present in RNA. So, if you use this, only DNA replication, during DNA replication this will be picked up. RNA, for RNA replication this will not be chosen because it is not present, it is not the monomer for RNA. So, therefore, this should be the correct solution. TTP with a labeled phosphate you should use if you want to selectively label the DNA. CTP is present, cytosine is present in both DNA and RNA. So, this cannot be used. Uracil is present in RNA, not in DNA. So, if you use this, this will label the RNA and not the DNA. So, this is also not correct. So, you have to use DTTP, then you can label only the DNA target or only the amplified DNA sample specifically or selectively. Question 4 is you have DNA and RNA. Question is, if you treat DNA and RNA separately, with concentrated sodium hydroxide, then which will be stable. Is DNA be stable in sodium hydroxide, or RNA will be stable in sodium hydroxide, or none of them will be stable in sodium hydroxide? So, this is base, so this is your DNA without the hydroxyl. I am writing the phosphate P, and then this is the RNA with a OH here. This is your RNA. If you treat this with NaOH, what will happen? If you treat this with NaOH, what will happen? The fact is RNA will be degraded, or RNA is not stable in sodium hydroxide. On the other hand, DNA is somewhat stable in sodium hydroxide. The reason is you are using a strong base. Sodium hydroxide is a strong base, and that will deprotonate this OH into minus, that you do not have here. So, phosphate is there, it's the rest of the part. Now, what this is going to do? This becomes a very good nucleophile now, very reactive nucleophile also. This will immediately react. This reacts here and it basically cleaves the other phosphodiester bond. So, rest of the part of the RNA would be cleaved off, and the first thing that we will get is phosphate, and there is nothing else here. Here as usual you have the other part, then followed by the other reactions, this will also be cleaved. But initially, yeah, here your extension, the other part of the RNA would be chopped off, and that will be happening for every phosphate group, for every phosphodiester bond that you have. So, every nucleoside positions or nucleotide position would be, or the backbone would be broken up or be destroyed in RNA if you use sodium hydroxide, because that is a very good base. So, RNA will be degraded or not stable, DNA would be stable. Question 5. In which medium this is about the protein structure, alpha helix formation would be favorable. In which medium alpha helix formation would be favorable? Alpha helix means alpha helix of the protein. A, aqueous, or B, lipid. Formation of alpha helix would be favorable in aqueous medium or in lipid medium. This is the question. What is the answer? So, when we talk about alpha helix, how is it formed? Alpha helix is formed by intramolecular interactions. This is formed by intramolecular interactions. Primarily the intramolecular hydrogen bondings. And intramolecular hydrogen bondings basically means the bond between the backbone carbonyl and the backbone NH. This is the hydrogen bonding. This is the hydrogen bonding, or there can be like this. I am just writing H here, NH in the backbone, they will form hydrogen bonding. And it is because of this hydrogen bonding that forces the strand to go to the helical conformation, because that way the backbone carbonyl and the backbone NH can come into the bonding distance. That is the driving force for the formation of alpha helix. Now, the question is, in which medium this should be favorable, aqueous or lipid? So, which is the other form? Other form is the beta. If the beta conformation is formed, then that is because of, as we have mentioned, intermolecular hydrogen bonding, NH, CO between two molecules, CO, NH, and like this, that will allow the formation of a planar structure. So, within a molecule, what will happen? If you keep it in aqueous medium, then the chances is high that your polar groups NH and CO both are polar, they would want to be exposed towards the aqueous medium, that means towards the outside of the backbone, and that will increase your chance of beta sheet formation. On the other hand, if you put it in a lipid, lipid is insoluble, lipid does not have water, lipid is hydrophobic. So, the polar groups would want to hide away from the surrounding medium. And how can this hide away? This is one good way, that they can have intramolecular hydrogen bonding, form a kind of bent structure which will be away from the outside environment. So, chances of getting the intramolecular interaction would be better in the hydrophobic medium, in this case the lipid. So, alpha helix formation would be more favorable in the hydrophobic medium. Just the reverse for beta sheet, this will be favorable in water medium, aqueous medium or hydrophilic solvent. Question 6, another question about the peptides is, suppose you are synthesizing, we will talk about the synthesis of the peptides later, but this is a very common thing. You are synthesizing a dipeptide. Starting with this is mainly for organic chemists, D and L, a mixture of D and L basically. Usually the common alanine is will be L conformations in protein, but here we are using both D and L, a mixture of D and L alanine, ALA for alanine, and a D and L of leucine. Then the question is, how many stereoisomers will you obtain? That is the question. If you form a dipeptide between alanine and leucine, and both of them have the 2 optical isomers, then total how many number of stereoisomers that will be having. So, if there is a dipeptide between alanine and leucine, then how many dipeptides can be formed? So, this can be the N-terminal alanine reacted with the C-terminal, reacted with the leucine and having a C-terminal end free. This is N-terminal, this is C-terminal, which means very quickly I am just writing ALA here, which just writing leu, because alanine and leucine none of them have any functional group side chains. This is acid, this is amine. So, if you react here, then you have the amine free for the alanine, which is called N-terminal, and your acid will be free for leucine, which is called the C-terminal. Now, it can be the other way around also. So, one is this, other one is leucine and alanine, which means the N-terminal in the leucine and C-terminal in the alanine. So, these two are the dipeptide kinds that will be in your solution, in your product. Now, you have to look at the stereochemistry. You have started with D and L both, also for leucine you have started with D and L both. So, if this is D, this can also be D, I am writing D prime, this can be D, this can be L. Right, this can be L, it is the same thing. No, it is not the same thing. L, this can be L prime, here all will be prime, this can be L, this can be D. 4, same here, D, D prime, D, L prime, L, L prime, L and D prime, and all of them will be stereochemically active, because these are two different molecules. So, if you remember your basic organic chemistry, if you have two optically active centers, the total number of stereoisomers that is possible is 2 to the power 2, 4. So, 4 for each, 4 for here, 4 for there. So, total 8 number of stereoisomers that will be possible for this dipeptide. Now, another very quick question is, you are doing the same dipeptide. I will not write the whole question. You are doing the same dipeptide, but with L alanine and with L valine. Then question is, how many optically active compounds will be formed? So, very simple answer, alanine. So, here you have only one stereoisomer of alanine, only one stereoisomer of valine. So, your dipeptide can be N alanine with C valine, or you can simply write alanine valine, and other case you can write valine alanine. This is conformation is locked, or the stereochemistry is locked L, this can only be L, D, D not possible because you have not used them, here also L, L. So, how many optical compounds total? There will be only 2 compounds or 2 dipeptides, both optically active. This is very easy. So, question number 8, a very interesting question actually. Which amino acids could be hydrolyzed to a different amino acids? So, if you look at the structure of the amino acids, from there you have to look at the compounds, or you have to pick up the amino acids that you can hydrolyze in water and can get another type of amino acid. Can you think of it? This is your amino acid. So, there are 2 actually of such type, CONH2. This is which amino acid? This is your asparagine, in short ASN. So, the amino acids which have a amide side chain, which have a functional group or amide functional group in the side chain, and amide can be hydrolyzed into acids. If you hydrolyze it in water, or if you use a little bit of proper condition, optimized condition. So, basically aqueous medium, it is a hydrolysis. Then what do you get is, water will attack here, this will go, this will come back, and eventually the amine will be eliminated. So, here you will get a OH, COOH, this amine would be there, plus you will release ammonia. So, elimination of ammonia would be there, and this amide group will be replaced or would be hydrolyzed into a carboxylic acid group. Now, this becomes another amino acid. This is your aspartic acid, ASP. That is one example. Second example is also having the same amide bond. Two CH2 and then the amine. This is your glutamine. GLN is the abbreviation. If you treat it with water, it eliminates ammonia. Now, you get another acid, carboxylic acid here as usual. So, this is your another amino acid called glutamic acid. GLU. So, I will write another question and I will leave you with this. Write the correct ionic form of the peptide which is given as N-terminal alanine, lysine, arginine, aspartic acid, and this is the C terminal. So, what will be the correct ionic form at pH of 7.4 physiological pH? What should be the proper structure of this peptide? We can do the I will show you the answer in the next lecture. Thank you.